Hello, everyone. Welcome to Math Bio Seminar. Today we have Ivy Xiong from University of Southern California. She does, I believe, a lot of systems biology stuff, including quantitative biology, computational biology. So thank you for giving a talk for us. Let's welcome her.
Thank you so much, Kana, and thank you all for the invitation. I'm glad to kind of share my recent study with you, which is titled A Common Pathway to Cancer, How the Oncogenic Mutations Abolish PV3 Oscillations. So I want to start the talk with kind of acknowledgement, because this is kind of a good combination of molecular biology, experiment on one side and computational modeling on the other. So I come from a background of biochemistry and molecular biology. And then during my PhD at Oxford, I joined the Bond Lab, which is looking at the the PV3 biology. I had the chance to kind of share, the first part of the study today. And then on the right-hand side is Professor Alan Garfinkel at UCLA, who is kind of expert in applying nonlinear dynamics to answer key biological questions. And I want to kind of thank the funders for the generous sponsorship during my PhD back in Oxford. So kind of the talk is divided into two parts today. The first part, mostly like evidence from molecular biology, is to look into germline by somatic interaction in the PV3 tumor suppressor pathway. So to give you a few background, I'm interested in cancer biology in my PhD, and the key, kind of lesson we have learned over the last 50 years is how cancer is driven and shaped by somatic mutations that occurred in kind of the tumors, cancerous tissues. My question of research during my PhD was to look into, does the inherited genome ever influence tumor evolution? So we know that the clonal expansion was driven by, hotspot mutations in one clone, but does it matter what the inherited genotype is of, those clones? So to answer this question, I have examined the most kind of mutated gene in the whole genome, which is the PV3. So a few kind of background into PV3 biology. So PV3 is a transcription factor that is activated by an array of stress signals, including DNA damage, hypoxia, oncogenic activation, oxidative stress, and telomere attrition. So when it's activated, it does a few different functions, including cell cycle rest, DNA repair, and apoptosis, which is commit cells to cell death. So my kind of central question is, can we identify some genetic, inherited mutations in the genome that would interfere with the somatic mutational status in PV3? As I mentioned, PV3 is the most mutated gene in different cancer tissues. I think in some cancers, up to 70% of the tumor samples will mutate PV3 somatically. So if you look at different mutations, we have identified across the cancer tissue. So this is from TCGA data. There's one thing that I found very interesting. So PV3 is known to be like a tumor suppressor. So tumor suppressor usually are just inactivated through deletion, or there's kind of loss of function mutation. However, if you look at the array of mutation found in within the PV3 gene body, I realized that a majority of them actually are missense mutations, fringe-shift mutations, which actually will still be able to retain the whole gene body. And then to kind of put it in another way, is like the typical mutation, which inactivates the tumor suppressor, we call it loss of function mutations. However, when you look at the function of individual mutations, I learned that PV3, in the case of PV3, a lot of mutations are actually doing something else, which include dominant negative effect, where the mutated copy would still bind to the wild type copy, but inhibit it from binding to DNA, and there's no transcription anymore. And another type of mutation is so-called gain of function mutation. So instead of losing its tumor suppressive function, the gain of function mutant now would actually go on to promote cancer instead of suppressing it. So this is more like kind of, so this two class of mutation would turn, PP3 from a tumor suppressor to more like an oncoprotein. And if you look at the distribution of, such mutations across different tumor samples, I found that 85% actually, kind of conferring oncogenic activities to PP3. So that kind of inspired me to look, across the genome to see that whether there are genetic variants that are inherited that might, interfere with this somatic mutational status, either, being a tumor suppressor or being a oncoprotein. So I did find out there's one variant. I'm going to just jump into that for your kind of narrative. So this variant is a to C mutation in the population. So this mutation or genetic variant is, located in the 3' UTR of the PP3 gene body and is in the polyadenylation signal sequence. So this minor C is present in about like 2% in the Caucasian European population, but is up to 5% in African population. So one thing that is interesting about this variation is that when you have the C allele, it seems that the PP3 expression is downregulated by twofold. So this is the case in both blood and in tumors. And you can also model this variant in a haploid cancer cell, a haploid cell line, basically like a cell line carrying either A allele or C allele. And again, we're seeing that PP3 expression, quantified by mRNA level is down by twofold. That's also the case you look at protein levels. So this variant, it turns out to actually interact with the PP3 somatic mutational status. So here is a kind of a complex plot, but I'll walk you through. So we have gathered a large case control study looking at breast cancer and ovarian cancer risk. So for the cases, it's nice that there are different molecular subtypes. For example, the ER-positive breast cancer versus ER-negative breast cancer. And then for ovarian, we have low-grade serous ovarian cancer and high-grade serous ovarian cancer. So one thing that I found is very interesting is that this different subtype of cancer actually have kind of a diverging phenotype in terms of whether they mutate PP3 or not. For example, like the one highlighted in red tends to mutate PP3 in the most majority of the cases, but like the subtype highlighted in blue tends to kind of retain a wild type copy. And when I look at the risk association profiles of this variation with cancer risk, I realized that if you carry a mutant PP3, this variant actually tends to be protective when you look at their odds ratio profile compared to tumors that has wild type PP3. So this is kind of suggesting there is a mutual antagonism between this genetic variant and the somatic mutational status of PP3. And we validated this by looking at cases in the TCGA cohort, stratifying tumors based on their mutational status. And again, we find there is a trend of correlation. So this variant C is enriched in cancers with Y-type PP3 and no copy number loss. And one thing we found even like intriguing is this mutual antagonism actually is also related to cancer prognosis. For example, here I take the breast cancer cases in the TCGA cohort and look at their survival curve over 6 years. So if you're just looking at the profile of genetic variant, it doesn't seem that the patients would have a different survival profile. However, when you do stratify the cases based on both the inherited genotype and the mutational status, we start to see a pattern. For example, we have the Y-type cases with the C and NEO. We know that this is actually a high risk profile. The survival profile is much worse when you compare to any of the other genotype combination. And what is interesting is if you look at the cases with mutant PV3 and the minor C, is actually having a superior survival and prognosis over time. So this kind of suggests this mutual antagonism is not something, just statistic noise. It's actually quite functional, and it's a real biological signal. To summarize the first part, I kind of showed you, like by this case study, looking at this variant, the PV3 mutation, I showed there's a mutual antagonism between the inherited variant and PV3 somatic mutation. So here's the story. If you have the major neo A, you'll end up transcribing PV3 as usual, and then you have abundant copy of PV3 when it's activated. However, if you carry the C allele, you end up having a lower level of PV3. If PV3 is wild type, that means we have less tumor suppression, therefore increased risk. However, if somehow like the tissues have mutated PV3, that would actually decrease risk by reducing this kind of oncoprotein expressions. So basically, like this suggests, depending on your inherited genotype, the probability of mutating PV3 during tumor development is kind of having a different level. For example, the C allele would tend to retain the Y copy, but A allele would tend to lose PV3 through somatic mutations. So this kind of have very, I'll say, interesting observation, because when we are looking at PV3 deletion, which is similar to the case of this genetic variant, we don't see this mutual antagonism. Usually we see the two actually co-occur. That inspired me to the second part of the study, which is the paper I'm sharing with you today, is I'm trying to find a way to unify this observation. Like besides the mutual antagonism, how about the other mutations in the PV3 pathway? Is a way we can unify them mechanistically. So that's like the second part where I want to show you that all these oncogenic mutations actually abolish PV3 oscillations, and that's how they promote cancer. So as I mentioned, the PV3 pathway has several actors. So besides PV3, we have P21, MDM4, ATM, MDM2, and the others. In particular, ATM is an upstream regulator of PV3, and then MDM4 and MDM2 actually regulate PV3 kind of by inhibiting its expression. P21 is a target gene of PV3. So here I'm showing you the mutational profiles of 7,000 tumor samples. So each kind of a vertical line here is an individual patient. By showing you their mutational status across this array of genes, I want to point out that how different mutations tends to be mutual exclusive, while the others tends to co-occur. For example, like PV3 mutations and deletions tends to co-occur quite a lot in tumor samples. And besides that, mutations within the same gene, for example, PV3, would have a variety of flavors. For example, we are kind of very familiar with many of those hotspot mutations, like the 273 residue in the DNA binding domain of PV3. We know that when you mutate this loci, this locus, the PV3 would turn into a nasty kind of pro-cancer phenotype in cell line. So this kind of prompted me to ask the question, like, how can we unify those kind of diverse alterations in the same pathway? So that prompted me to look into PV3 dynamics. One thing is PV3 has been really well studied in the last 50 years, and the Hopf lab basically have showed how this PV3 MDM2 feedback loop coordinate PV3 dynamics in single cell over real time. So PV3 as a transcription factor, when it's activated by, for example, DNA damage, it would accumulate and transactivate the expression of MDM2, which is the inhibitor of PV3 activation. So there is a time delay here due to transcription and translation. So this negative feedback with time delay actually produce oscillatory patterns that has been recorded very robustly. So here I'm showing you a single cell, live cell imaging over time. So if you kind of trace different fluorescent signals in PV3, which is in green, and MDM2 in red, you can see there's kind of oscillatory pattern that can last for several days. So the Hopf lab at Harvard Medical School, so they characterize this oscillatory behavior to have a period about 5 to 6 hours, and they have proposed multiple mathematical models to try to explain this dynamic feature. Another
Very quick question.
Oh, sure.
Does the period vary depending on cell line?
So it depends on species, but not cell line.
Oh.
Yeah, they've done a really great survey. Like if you take normal cell line in human, if you take normal cell line, cancer cell line, they all have the same characteristic period. However, if you look at mice, it's 3 hours.
Oh.
Yeah,
Cool.
I think primate dogs, I think, has something in between. Yeah, there's a really great paper by Hopf as well.
Thank you.
No problem. Another question I had is, okay, now the oscillatory behavior has been well recorded and it's very robust, but what does it do? It turns out it's not a straightforward question, because many people in the field are kind of, having a strong resistance to assign any functional role to PV3 oscillations, as well as other transcription factors. However, by surveying like recent literature, I did find something. For example, if you look at PV3 oscillatory patterns and how it's sustained cell cycle arrest, I found out that if you see a switching behavior, so you switch from oscillatory pattern to a constant high level or a high dose, the cells would escape from cell cycle arrest. And then the Hopf lab back in 2012, they characterized this oscillatory behavior against the constant high level PV3 and to see how the cell tends to behave. It turns out that if you have a constant high level PV3, the cell will commit into senescence or apoptosis really early in time. For example, here, the T1 and T2 is real time. So instead of commit to senescence or cell death at a later stage, having constant high level PV3 seems to be toxic. It will just prompt the cell into cell death or senescence right away. Therefore, I learned that PV3 oscillations are quite essential for cell cycle arrest and DNA repair. Therefore, I reason like impaired PV3 oscillations would compromise genome integrity and promote cancer. This is through like kind of accumulation of DNA damage that is not repaired, but cell progress into cell cycle and amplify into their daughter cells. So I kind of took one of the models that Hopf lab have characterized through like real experimental data, and then trying to see how different mutations in the PV3 pathway would impact the dynamic behavior of the system. So here I have a 3-variable ODE model, where the 3 variables are quantifying PV3 protein levels, MDM2 transcript, and MDM2 protein levels. So this is the same negative feedback loop that is, having time delay built in through transcription and translation. So this simple mathematical model kind of recapitulate a few of the biophysical features of this circuit. For example, PV3 is produced with a constant rate alpha and degraded through binding to MDM2 with a Hill function. MDM2 transcription is induced by PV3 through a Hill function again, and is degraded linearly. And the protein of MDM2 is produced proportional to the transcript level and decay linearly. So I have parameterized this model, which produced this kind of nice 5 to 6 hour characteristic period in all 3 species. And then that's also shown in this state space trajectory, looking at the limit cycle behavior. So it doesn't matter where you're approaching the limit cycle, it will converge in the end. So the first thing we can do is that now we have a mathematical model, specifying PV3 dynamics that has the oscillatory features. There are a number of things we can do. The first thing I did was looking at how PV3 can coordinate cell cycle arrest and DNA repair versus apoptosis. So there are 2 kind of target genes to represent the 2 outcomes. So the first is P21 that is activated to arrest cell cycle, and the other one is PUMA, which activate BAX and induce apoptosis. So these 2 target genes of PV3 has been characterized through biophysical studies. So I just took the parameter values and simulated the expression profile. For example, in normal response, we have oscillatory PV3 dynamics and how PV3 would induce P21 really efficiently. However, PUMA seems to have this kind of pulsing counter behavior that accumulate much slower over time. So this might explain, the normal response to moderate DNA damage, which is you try to arrest cell cycle and facilitate DNA repair. However, if this signal persists, which means DNA damage is still not repaired, the accumulation of PUMA will commit cell to apoptosis at a later stage. And then we can also kind of looking at how different response, other than normal response would impact on the 2 outcomes. For example, when we have PV3 deficiency, basically we reduce, the production of PV3 by 1/2. We realize that it cannot induce P21 efficiently, and PUMA doesn't really accumulate. So that's actually recapitulating what we know about the biology. So when you lose PV3 even by 1/2, the system will not be able to cope with the challenge. And another scenario is acute PV3 activation through MDM2 inhibitors like NATLIN3. We see a potent induction of both P21 and the PUMA and commit cell to apoptosis immediately. So that's really early on in the response.
Sorry, I just want to ask something I think I missed. So I understood the model that you proposed for P53. Here, did you now couple the P53 response to both P21 and PUMA? Was that on the previous slides? I think I just missed this coupling.
Yeah. So here I'm kind of using MDM2 in the main model, but the other targets are actually the same. The only difference is, for example, P21 does not feed back on P53. So like in the paper, I actually singled out, target genes that not feed back into P53, but they have the same form.
I see. So you parameterized those in the same manner.
Yes.
But then I guess one question about the fitting. Did you, when you were doing the parameter fitting, did you use the same parameters that you had fit to the MDM2 response, or did you use like new fitting curves? And if so, which one is kind of the fit and which one's the prediction?
Right. The original model was well parameterized by the Hopf lab. So I, only modified it a little bit. And then the other target genes actually was measured biophysically, like the parameters, by a postdoc lab. Like, yeah, so it's related. So they have the same.
Those are actually measured as well.
Yeah, yeah. Okay. So he, I think it's bachelor. They are so he's at NCI right now. So he did a bunch of those biophysical measurement of the same system but looking beyond MDM2. So
I see. Okay, thanks.
Yeah. So that was really helpful. I don't know about other systems. Maybe we need such biophysical measurement before we can parameterize in the system in this consistent way. So taking this model, now I start to survey all those different mutations I mentioned before, plus a number of more that I didn't mention. So here is basically a summary of what we have learned from molecular biology evidence, like about PV3 biology, how different mutations that impact different biophysical parameters. The table is pretty big, but I want to point out how this inherited polymorphism reduce the production rate of PV3 alpha. And then we have also mentioned PV3 somatic mutations, including the Hopf mutations, which reduce the inhibition term beta between MDM2 and PV3. And there are also other missense mutations that reduce sigma, which is the transactivation rate. So that's the transcription activation rate of MDM2 transcript. So besides that, I also noticed that there are 8 parameters in this model, but most of the mutations I've surveyed are only impacting the 4 parameters I've listed here. So I've mentioned the production rate of PV3 alpha, and then we have beta, which is the inhibition rate between MDM2 and PV3, and then transactivation rate, sigma, and delta, which is the degradation rate of MDM2. One thing I want to kind of point out before we move on is the same mutation can influence multiple parameters. For example, loss of ATN would increase beta, but also decrease delta. And then different mutations can pull the parameters in very different directions. For example, Hopf mutations decrease beta, but the others, for example, loss of ATM or overexpression of MDM4 actually increase beta. So it's kind of a whole array of behaviors that's how different mutations, impact on the behavior of the system. So my question here is, can we actually find out like what all those mutations are doing to the dynamic feature of PV3? So I started by looking at alpha, which is the production rate of PV3. If we reduce it by a half, as we have seen earlier by PV3 deficiency, the time series or the tracings of PV3 tends to show a damped oscillation behavior, and that we know that this is not essential. This is not sufficient for tumor suppression. And you can also look at this time series, the tracings for different parameter values of alpha. So here I'm showing on the x-axis the time, and then the y-axis is different parameter values, and then the value of PV3 is shown by the heat map in red color indicates high values. And then what is interesting is if you look at like this, reducing alpha by a half, now we start to see kind of loss of oscillatory behaviors by losing of this waveform. We can also do that for the other 3 parameters. So delta seems to be similar to alpha. When we reduce delta, the system loses oscillatory behavior. And if you look at beta and sigma, the system seems to have kind of reverse phenotype. For example, if you reduce beta or sigma, the system actually have a slightly heightened oscillatory behavior, and then you end up losing it when you reduce it to a much, much lower level. However, if you increase beta, the system also lose oscillatory pattern. And then for these individual parameters, you can do bifurcation analysis on single variables by plotting the steady state oscillatory, the maximal and minimal values of PV3 against the equilibrium point at the other parameter values. So by examining this bifurcation diagram, we kind of start to see there is some kind of bifurcation going on. And then we check the literature. There are different bifurcations that can give you such behaviors. But by examining like this kind of almost like a set node behavior, we kind of suspect the systems going through a Hopf subcritical bifurcation. So this prompts us to actually looking at the system in a totally different way, which is, can we actually call out that there is certain qualitative changes? By saying that, is like we have looked at the time series at different parameter values. We have examined the bifurcation diagrams, but can we actually study the bifurcation analytically, changing from having a steady state behavior to a robust limit cycle behavior and come back? So that kind of brings me to the Hopf bifurcation theorem, which is a way that we can actually study this bifurcation behavior analytically. So the theorem basically says if we look at the eigenvalue structure of the equilibrium point, if the pair of complex conjugate eigenvalue passes the imaginary line, as the parameter passes the critical value, then the equilibrium becomes unstable. And that's how a limit cycle chapter can be born. For example, here is a toy example. We have parameter value C. So as we decrease C passing through the critical value, the system change from having a steady state behavior to a robust limit cycle behavior. And then the corresponding eigenvalue structure shows it changes from having a negative real part to a positive real part. So this kind of inspired me to quantify the PV3 system in a similar way. So to do that, first step is to find out the equilibrium point of the system. So we have a 3-variable ODE system, so we can solve it analytically or symbolically. So here's kind of a messy formula, but in the end, we have all 3 equilibrium point calculated symbolically. The second step is to perform linear stability analysis, calculate the Jacobian matrix and find out the eigenvalue structure. So this eigenvalue can be solved through this qubit equation. If we kind of annotate P and Q to represent this large complex expression, we can actually simplify the 2 potential complex eigenvalues by this form. And then it's quite obvious to find out the Hopf criteria for sustained PV3 oscillation. So basically the P, which is the expression up here, has to change from being positive to negative. So now you will have the positive real part. And of course, the part under the square root would be negative, so the imaginary part exists. So using this kind of set of criteria, now we can examine the system and perform multi-variable bifurcation analysis. For example, we can actually combine all 4 parameters that are perturbed by somatic mutations and inherited mutations, and then see how it affects the behavior of the system. So here is a kind of 3D iso surface plot. So this gray surface represents a Hopf boundary. So inside the surface, so the area enclosed by the surface represent when the system would actually have robust oscillation. And then outside of the surface would represent regions that have no sustained oscillations. So the red dot represents the best fit parameter value. So that's the parameter values I used to simulate the system at the first place. And then by looking at the changes of different parameter values along the axis, now we can find out how different mutations actually all pull the system out of the Hopf bifurcation, like the area where it has oscillatory behavior. For example, loss of PV3 would pull the system down and out of the Hopf region. PV3 missense mutations will pull the system to the left along the arrow here. it's a long way, but in the end, you'll be out of the oscillatory region. And then overexpression of MDM2 to MDM4 would put the system to the right, kind of diametrically from PV3 missense mutation, but it also put the system out of the oscillatory region. Loss ADM kind of put the system sideways, but has the same outcome. So now actually we can explain why we saw the mutual antagonism in the first part. So the genetic variant I've introduced in the first part actually pulled the system just a little bit. It's about a quarter decrease. So now it's kind of done by 25%. So it's kind of marginal. If you mutate PV3, which will push you to the left, it will actually bring the system back to the oscillatory region. That would actually hinder cancer development. So that's why we start to see the mutual antagonism and has also a association with prognosis. However, if you start with the Y type or like the major allele A, you want to lose PV3 all the way to the left, so the system can become cancer in the end. So you can actually rotate this object and then see how the system would behave differently due to different mutations.
I had one question. So in one of the previous slides where you were showing the role of P53 in tumor suppression, one of the behaviors was kind of the sustained oscillation. And I guess another one that you showed was the switching. Does this also capture the switching, or is that in also some different regime?
Oh, the switching, I think it depends on the time. So what I did is they used I think an inhibitor to kind of coerce the system out of the oscillatory region. So it's not reflected through the bifurcation diagram, like the 3D diagram I've just shown you, because that's actually looking at the steady state or kind of the long-term behavior.
I see. Okay, thank you.
Yeah, I can send you like their paper. I think they did a good job kind of characterizing the switching behavior as well. Yeah, so here is kind of come to the summary of the second part. So I've shown you how in response to DNA damage, PV3 normal physiology oscillate to facilitate cell cycle arrest, DNA repair, and in the end, the cell will recover from the damage and challenge. However, due to oncogenic alterations, the whole range of behavior now actually lose the oscillatory pattern that leads to accumulation of DNA damage and induced instability that kind of precedes cancer development. So besides PV3, which is mostly in the category of cell signaling, we also have NF-kappa-B oscillation. There are actually a range of other functions those physiological oscillations serve. One thing that was chatting with Kana is how has one in the segmentation clock keeps biological time. But there is an emerging theme of how oscillations actually can synchronize couple processes, which is widely seen like in circadian rhythm, for example, but also in a whole array of metabolic oscillations, how it facilitates, for example, insulin release in the pancreatic beta cells. So this is something that is upcoming. So I would love to share with you another time. Yeah. So another thing I want to kind of advertise to you all is there's one little thing I'm organizing, the Dynamic Club at USC. So we are hosting like a range of diverse topics throughout the year. And our our first guest lecture is going to be next Thursday. So it's going to be 11:30 at Eastern Time. And we'll have two speakers, Dagmar Iber from ETH Zurich, and Dennis Menschikow to talk about pattern formation and also insight into industry, how computational modeling can be used for pharmaceutical development. So everybody is welcome. You can find out the information on the web, or I mostly do the announcement on Twitter as well. And I'm also recruiting speakers for next year, 2023. If you are excited to share your latest research, please tell me. I would love to host you either on Zoom, or if you happen to find a way to LA, I'll invite you to USC. Yeah, so thank you so much for your time, and I would love to take any questions.
Yes, I had a question about the type of Hopf bifurcation that you have. So it seems that you have a subcritical Hopf, right? Creating unstable cycles and then a fold
Yes.
of cycles. And on the other side, is it the same, or I mean?
Yeah, all four are subcritical of.
Okay. And all right. And does that make any difference for the phenomena? I mean, I guess the main difference is by stability in the region between the Hopf and the fold. And also the fact that if you had a supercritical Hopf, you can also generate very low amplitude oscillations, whereas here they directly have large amplitude. So I don't know if it's...
Yeah, like we actually had a little discussion about supercritical versus subcritical. So like when it comes to PV3 biology, I think it would be beneficial for the cell, like my reasoning is to have kind of fully blown oscillation immediately instead of going from like tiny to the large oscillations.
Okay. And did you check the type of Hopf that you had, or?
Yeah, so we kind of examined by like, are there actually bistable regions? We did find that such, especially here.
Yeah, mathematically you can compute the second term in the Taylor expansion of the normal form, but I don't know if you did that or.
Oh, for the Taylor expansion, no, we haven't done that.
Okay.
Yeah, I think that would be interesting to look as well.
And in the surface that you have, I was wondering if you had a systematic way to restore oscillations by increasing alpha or not, or if the region is, I mean, I'm wondering, essentially my question is, does it go all the way to infinity, the surface, or is it, if you project it on the delta beta space or not?
Oh yeah, we had one part. It looks like there is an island. So for example, you take like cross sections of this isosurface, is not going to infinity. Like we call it the Goldilocks effect. So like you have to be somehow in the sweet spot to have the oscillation. And then one thing like to keep the system in check is PV3 does not oscillate on its own. It needs activation signals. So ATM in response to DNA damage would come in, ATR as well.
Yeah.
So yeah, it's not spontaneous.
I guess my question was, if you increase alpha, you don't necessarily go up to oscillations, right?
Oh, you mean the alpha here? Yeah, I think I can look for a bit bigger range. Oh yeah, there's this case, like if you think about how like this is human PV3, well, we have one copy or two copies of PV3. For elephant, actually have 50 copies. So that might means that their alpha is much higher. Well, that might like people reason like that's why they have better strategy to counteract cancer. Yeah, that would be a good argument to test as well. Like when you have sufficient large alpha, where the system is more robust.
Yeah. So and that's why I was asking, your surface is kind of like that. If it goes all the way to infinity.
Oh yeah, you mean on
from
top?
Any point, going up with the alpha would eventually cross the surface. And so you could think, well, it's just to restore, cellular function, I just need to increase alpha enough. give a drug that would do that. But looking at it, I don't have the sense that I don't know, in fact.
Right. So like you're suggesting I can elongate the surface by going up like in alpha from
Yeah. And will
Yeah.
you always cross the surface when you go up,
Yeah, that'll be nice to chat.
Yeah, I was thinking in the same line, because according to the 3D diagram, if alpha is bigger, the oscillation, the area seems bigger, and it will be more robust to many mutations.
Yeah, you're right. Like if we just move the red dot up, the tolerance of mutations will be much higher, right? Like because you will still be in the oscillatory region. Yeah, funny thing, like if you have the 3D, actually like there's a curved surface. Yeah, I think those are great hypotheses. Actually, like when I was telling this story to another institute, they was like, maybe you can test it already, like grab a cancer cell line in the lab. Like throw a
Yeah, is
check.
there a way to test that? To experimentally increase alpha in culture cell? Is there a way?
Yes, exactly.
That's so cool.
Mm-hmm.
I had another simple question. Why there is no degradation term for P53?
Oh, because so there is. Like depending on what model you look at. For this one, because it's the degradation through MDM2. So MDM2 is a
degradation.
E3 ligase. So there are baseline degradation, but compared to the MDM2 mediated degradation is just it's just negligible.
I see.
Doesn't make sense. Like E3 ligase tag PB3 send it to proteasome for degradation.
And I had one more question.
Sure.
If you can go to slide, I think 20. Was it 20? So according to these graphs, beta and sigma changes also the period of oscillations, right? Does that have any physiological function, like period change?
Yeah, so that's another thing that might interesting to look into. Like, does the period matter? Yeah, I had like one lines of research when I was finishing up is to looking into the periodicities, because my reason is, why does it need this characteristic period, right? 5 to 6 hours. Is it possible that PV3 oscillation is coupled to other oscillations in the cell? Yeah, but like we didn't find any experimental evidence for that. So PV3 is a very versatile transcription factor in the cell. So it's activated in like by an array of signals if you come back to the biological background. And it interacts with many different actors. So it's quite hard right now, like for me to figure out what the partners are. So we have been looking at HSF1, which is a heat shock protein as transcription factor for heat shock proteins. So that might be one way that the characteristic period is important. But yeah, we haven't really developed that line of research to its maturation.
Is there any synchronization you observe among cells?
Yeah, that's kind of how I'm hypothesizing about the characteristic period, but we don't really have a lot of biological evidence for that as of today. So it's hard to kind of make that argument right now. But like HSF1, I think like temperature regulated cell functions has been, kind of tracking a lot of the tensions recently, especially when it comes to circadian rhythms. Maybe in the near future, there will be more evidence coming up, and then we can just hook up PB3 with the other activators like HSF1 and maybe the MAPK pathway as well, to see the synchronization of PB3 action with the other processes like cell growth and heat shock response. I think that's quite interesting.
Thank you.
Thank you.
Do we have any other questions? All right, if not, thank you, Ivy, again for giving the talk. And I personally look forward to listening to your talk at Systems Bio at Harvard next week.
I'll keep you posted.
Thank you.
Thank you.
All right, thank you
you.
so much.
Thanks, Ivy.
And the rest of you, we will see you in 2 weeks. Okay, thank you. Bye.
Thank you.
Bye.